Today we're going to do a migration assay. I just took my plate out of the incubator. It was in there for 24 hours. You need to make sure you're wearing gloves and that you're not under the hood. This doesn't have to be sterile. So first thing we're going to do is take the lid off. Right here we have our SKS cell lines and here we have our A204 cell lines. First thing that we're going to do is vacuum off the liquid underneath the well. So you just lift it up and vacuum it right up. And next we're going to add 400 microliters of cell stain solution, which is in this bottle, into each of the wells above and below. Okay, and then the next thing we're going to do is vacuum out all the empty wells with a new pasture pipette tip. You just want to be very gentle. You don't want to press against the bottom of the well here. That's where all your cells are, and that polycarbonate membrane can be kind of sensitive. And you want to get a new one for each cell line. Okay, and now that that's done, you're going to take a Q-tip and you're going to rinse it with PBS. And this is 1X PBS. So once you're done rinsing it, kind of press it against the sides because you don't want a whole lot of PBS on the Q-tip at one time. And you're going to wipe it against the inside of the well. You want to be kind of sensitive. You don't want to puncture this membrane at the bottom of the well. You just want to be really thorough and try to get all the cells that could be like not stuck inside. You want to get those washed off. And you can use the same Q-tip more than once. Okay, and you're going to want to switch the Q-tip use for each cell line as well. So I'm going to use the other side. Okay. And then we're just going to put these each into the cell stain solution all at the same time. Okay, and now it's going to go on the shaker for... Yeah, let it sit for 10 minutes. Okay, so now that 10 minutes have passed, we can take our wells that we have in here. We're going to grab them with these tweezers. If I can do it. Alright. Grab them with the tweezers, and you're going to rinse them out with the deionized water here. And just kind of run it through a few times, get all the stain out. set it upside down on the paper towel. Make sure it's labeled so you know which cell line is which.
Okay, so to speed up the drying process, carefully lift up the insert and vacuum the inside carefully.
Okay, so while they're drying, we're going to add 400 microliters of cell extraction buffer to each well. We're going to want to do 3 extra wells so that we can blanket with just the cell extraction buffer. Okay, and now we're just going to add our wells in there. And we're just going to put it on the belly dancer for 10 minutes. Okay, it's been 10 minutes, so we can put our plate in the plate reader to read it. We're going to take this vial and fill it with water. Make sure the pasture pipettes go in the glass container, and we're going to bleach any cell, any area in here